Hi, so this course is an introduction about population viability analysis. I will make 2 or 3 videos actually. This one is a presentation for you to understand why it's important, and then I will show you which software we can use, namely Vortex, and then I will do a second video about this software, how to use it, what parameters, etc., etc. But now let's do an introduction, because I want you to understand why it's super useful to know how to use Vortex, or why not another population viability analysis software. Because you, as biologist, as conservationist, you will maybe need to use this kind of tool to know what will happen to your population, maybe your species, or to your new population after a reintroduction, or maybe it can be a species which is an invasive species, and you need to know if it's too late, or if you can do something, or if there is a bioparameter where you can act to save your island from this species or something else. So I will do we'll do together some examples to understand why it's important and what we have to think about when you start to do a population viability analysis. So in conservation biology, it's quite useful, it's quite widely used. Obviously, you know why. Probably during your bachelor degree, you learned how to do a population model, how your population can evolve due to the number of babies the female will make, due to the mortality, etc., etc. We'll think about all these bioparameters later together. And you probably learned that you have deterministic models. It's when you say, okay, I have 3 females with 1 male, it's okay because it's lion. It can be only 1 male for several females. They can have 5 babies each female. The probability of survival is this amount for the first year, and then the next year it will be etc., 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 etc. Okay, and then you have an estimation of your population size, of your number of individuals year after year or after month if it's insect, fish, or I don't know, with a shorter lifespan. And you heard that it's not like that in the nature, because you can have fire, you can have storm, you can have a plague, you can have stochastic events that will change your prediction. And then it's what we call stochastic event that will change your model. So we'll work on stochastic models, okay? Because most of the time when you will follow a population, you know what is the amount of fire, maybe, or big drying event, things like that, and maybe hopefully you know what will be the impact on the survival, on the mortality rates, or on the number of baby, etc., etc., who will be produced. So now we work on stochastic models. So this is a deterministic model, plus we had some events, bad events, who will change the probability of survival of the young or adults, things like that. Okay, when you will use stochastic models, for example, it's if you have to manage a little population in zoo, for example. Actually, Vortex was made by biologists from zoo. Let's say you have 3 kangaroos, female, 1 male, according to the biological parameters of the species that you hopefully know. 1 female make a baby every 1.5 years, for example, with an average rate of survival of then you can maybe predict how many kangaroo you will have in 10 years, 20 years, things like that. Is it viable, this size of population? So first, it was made for biologists in zoo, for captive population. But obviously you can use it also for a little population of animals In the nature, for example, you have very little survival of a population on an island, and you want to know if it's viable, you can use this kind of model. If, for example, you have a tiny population and you have a number of individuals who are poached, you can also try to do a model to know how your population will evolve. Is it viable? The number of animals that are poached, or maybe just hunted, because a lot of population, unfortunately, are also impacted by hunting, legal, I can say, authorized, let's say, it's maybe better, authorized number of individuals killed, what we call quota. So, are this quota viable for your population? So, yeah, you understood you can use it for endangered species, little species that are submitted to various population parameters. But also you can use it for invasive species. For example, you as conservation biologist, maybe you will have to manage some invasive species that threaten your target species that you want to protect. I have some example here in Florida, or you have some predator on islands who can kill many seabirds that are not used to live with predators in islands. You know, rats, cats. Here, it's a carnivorous. But now you know that we have many problem of conservation due to invasive species. Okay, so you as conservationist, maybe you will have to deal with this kind of problem, and then maybe you can do a viability population analysis, not on endangered species, but on invasive species, to predict how your population will increase or decrease. If you want to compare different scenarios of management, okay, like targeting the female or the baby. Also, it's good to be able to do population viability analysis, because when you are an expert on a species, maybe you will have to deal with the status, the red list status of the International Union for Conservation of Nature. You know, we'll do maybe a course together about the red list, how to understand the status and how you can change, upgrade the status of a species. Actually, one of the status upgrade can be from a model, a mathematical model, which is criterion E, because there is several criterion A, B, C, D, E. I can show you. It's here. Here you have the summary, actually everything, but I will show you the summary. Where is the summary? So you can download the summary. Show me the summary. Yes, here. You have the summary about the criteria to classify a species according of the population size or trend. For example, reduction in geographic distribution, okay, it's criterion A. But look, criterion E, the last one, here. It's about quantitative analysis that estimates the probability of ecosystem collapse. So it's exactly what I will teach you right now to you. If imagine you have a population, a species that is well known, you know how many female you have, what is the probability of survival, etc., etc. And if you make some quantitative prediction, and you can show that within 50 years we'll have 50% less individuals, for example, you can turn on the red light. Hey, I have a problem, my species is probably critically endangered. So you see, it's good also to be able to make some predictions, models about your species, because also for status, IUCN status for the red list. Okay, let's go back to the presentation. So now you know, as a biologist, why it's good to be able to do population viability analysis. Again, I want you to think about, now I know why it's important, but what I need to do a population viability analysis. You probably worked on it, okay? But let's dive again into your memory from your bachelor degree. Let's say I have 4 examples with 4 species and 4 scenarios about conservation, okay? And I want you to think about, what do I need to do my model, okay? My population viability analysis. Let's say first you have the Asiatic lion. You have to manage this population. This population actually is in a very bad shape. 1 millennium ago, this species was still in Greece, in Europe. You know, all the legends about lions. It's actually because the lions were here in Greece. They made some statue about lions because lion was here. It was part of their environment. I mean, a bit into the wild, huh? But it was a species in Europe. But nowadays, it's only a population of 100 of lions only in India. You heard about maybe the Gir National Park. It's a place, the last place for this species. In the 50s, they were also in Iraq or Iran also, yeah, Iran last. They were also in Iraq before. So now it's only in India, and only 100 of individuals. So let's say you have to manage this population. Actually, now this species, it's only one population. Do you think it's viable? Not think, but you as a biologist, can you prove this is viable? Only 100 of individuals. So let's think about, what parameters do you need to make your models. Okay, so I want you to make pause and write, what do you need. Then let's say we have another examples. I show you 4 examples because I want you to think about different scenario about conservation, because it's not the same when you want to do a model for lions and when you want to do a model for birds. Monogam, maybe. So indeed, let's say now you have to deal with a bird which is endangered, critically endangered. This bird, Pomaria, is only is a species that live only in a tiny island in Polynesia, in Wauka. I will show you. So monarch, it's here. You see, this is the male, this is the young, and it's only in this beautiful, majestic island, which is a tiny island in the Marquesas archipelago, not very big. And actually, this species, you have maybe 5 couples, only 5 couples and few young. And you can have, for example, the government who provide you money for your project, asking you, hey, is it useful to give you all this amount of money every year? Maybe we can spend it for something else. Can you prove it to us that it's still viable? So what bioparameters do you need to make your models? When I talk about bioparameters, it's how many young a female will lay. Do a female need a male? Yes. For the species, for example, you need to know it's a monogamous species, so you need 1 male for 1 female. If you have 4 female and 3 males, sorry, but you will have only 3 females able to reproduce in this case. I gave it to you as examples of bioparameters that you need. What else? So press pause and think about other bioparameters, all parameters that you need to build this population viability model. Another example. Let's say now it's not an endangered species that you want to study, but an invasive species. Let's say you are in the south of France and some hunters, some other people working into the nature told you that they found 3 raccoon dog. So raccoon dog is a species, I show it to you. Raccoon dog is a species that come from East Asia, but in the East Europe they took some of them to make fur for the fur trade. And obviously some of the individuals escaped, and now they are well established in West Europe. And slowly you have little pocket of population found, for example, in France. Let's say that you are here and you are a bit stressed, and the public authorities, and maybe also you and other scientists, want to know if it's a big red alert. Do your new individuals can make a viable population? So maybe you want to make a population viability analysis. What do you need? What bioparameters do you need for that? Now because you worked on the lion of Gir and you worked on the Pomaria from Wauka, this little bird, you probably already have the same parameters. Obviously, 90% of the bioparameters will be the same for all your models, but let's think about others. Okay, press pause, think about that, write it. And let's say the last scenario, you are a biologist working on marine mammals, and you know that sperm whales are still hunted actually into the world, not only by Japan, Norwegian or Iceland, but also in Indonesia. You have a little population of human in Lama Lera in Indonesia. I can show you a map. I have a map here. So you see, zoom out. Indonesia, it's a big country. So several islands. And here in Pulau Lembanta, you have a little community, few tribes here who are still allowed to hunt sperm whales, because traditionally blah 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 blah, and normally they have a quota of 15-20 per years, but some journalists say that some years they hunted 51 sperm whales, adult sperm whales. So is it viable for this population of sperm whales? Don't forget, as biologists, you know that whales are not it's not a big soup of individuals that can mix around the world. Quite often, same for fish actually, you have a population, a restricted population. Okay, they can have a big range, but the sperm whales from here are not the same population from here. So let's say that this population over there, they move a lot actually, sperm whales in the area. The big population here can survive with a poaching, hunting, annual hunting of 50 individuals per year. So in this case, if you know strong parameters about your population, which parameters? Do you think about that? You can predict if this rate of killing is sustainable or not. Okay, so I provide to you 4 examples of conservation where population viability analysis can be useful, can be an interesting tool for you to know which parameters is important, or to prove to stakeholders, government, or something like this, that you have to do something or you have to change something for your population. Okay, so press pause if you still need to write, to think about that. Okay, so I'll tell it to you what you probably wrote. Okay, so when you do this kind of models, you need to know if your population is monogamous, polygamous, if it's strict or not. What is strict? It's when one male can fertilize several female, even if it's monogamous. If one male can deal with 100 of females, this is the case, for example, for elephant seals. So you need to know that. You need to know how many female will reproduce, and from which years to which years. So what is the lifespan of your male? The lifespan of your female can be different. Obviously, it's not the same for a male lion and for a female. Not the same life at all. So until when they can reproduce, what is the rate of reproduction, how many babies a female will reproduce. For a sperm whale it will be 1 probably. For a lion it can be 5-6 cubs, depends of the population. You have to know that. And then what is the survival rate of the young? So for the first year, second years, etc., etc., until they are adults, they can reproduce. You will need to know the amount of adults who will die after a certain age. You need to know that. You need to know so yeah, at what age the baby are able to reproduce. Okay, you need to know what else. Tell me. You probably wrote that it's possible to put an amount of individuals that are extracted from your model. It's hunting or poaching, or if you do a program of reintroduction and you want to know how many individuals you can remove from your population, so you can add how many individuals are removed from your model, from your population. It's actually poaching, hunting, or extraction for conservation purposes. Or it can be because it's a natural migration from a population. So actually you can build a meta-population models, and you can explain how many, in Vortex, what I will show to you later, how many individuals migrate, okay, from one population to another, and maybe the rate is not the same from the little population to another. Remember, the population who are source and population who are wheel, just individuals going in but nobody escaping or migrate. Okay, so you need to know that the amount of migration for male and female obviously is not the same. For example, for lions it's mainly males who migrate. So for that you will need, 51 sperm whales killed, extract from the population. You need to know what else. You probably wrote now the stochastic events. It's very important for your models to know how many catastrophic events can occur during your estimation. You probably know, if you are the ranger of the Gir National Park, how many fire events occur every 10 years or whatever. The same for drought, for dry events, etc., etc. And then you can check what bioparameters are affected. if it's the survival of adults, the rate of young can fly, etc., etc. So you need this kind of bioparameters linked to stochastic events. Okay, I think we have mainly everything. Anyway, you will see when we will have to fill the models. If you forget something, you will have to check it, if it's available. What kind of software we will use? I will present to you a quite powerful tool which is free and can work on several platforms, okay, namely Vortex 10. Okay, so here I will show you the link of my presentation. You can click on Vortex 10, but you can type also vortex10.org and you will find the Species Conservation Toolkit initiative, providing several software, especially Vortex, the one that we want now. So you can click pause and have a look, download it, it's safe on your computer, check the last version. Actually, let's go and I will show you in January 2021 which one is the last version. And for the next hours we'll work together on it. Actually, what I will ask you during this course is to make your own model. Pick up your population, the species that you want, the best beloved species that you want. Some of you already know which one it will be, and I want you to show me if you can do a model. Not found. Oh, okay, Vortex. Vortex 10. So Vortex is also a word for Vortex. It's actually a picture about after a certain amount of individuals, you can have a Halley effect. You probably saw that effect, Halley effect. When you go below a certain number of individuals, it's doomed for your population. Because even if, for example, you have 10 individuals, adults who can reproduce, they don't have enough stimulation, for example, to reproduce or things like that. So sometimes you have Halley effect. It's actually something that you can put in your model. Halley effect, say below a certain amount of population, it's doomed. Okay, that's why the vortex, because sometimes less and less individuals, and then you have everything catched into a vortex of extinction. Okay, Vortex 10. So it evolved a little bit since my last download. Well, it's changing indeed. Okay, I will update the link. Vortex population viability analysis. Okay, as I told you, first it was produced by the Zoo of Chicago, and you download. Okay, you will have also the manuals if you need to go deeply in it. Okay, and c'est parti. Voilà. Okay. Now let's go deeper into the software. I will show you what you need to know and what you can put into your models to make it, and the most important, what can you do when you don't know about your population or your species, because obviously that's why we need biologists to follow, to monitor, to census a population. It's to have this kind of bioparameters. But when you don't have it, because obviously we don't have it for all population, all species, what can you do? And is your model reliable? We'll work on it for the next on the next video. See you.